Hello humans on this random planet called Earth, and welcome to my channel. My name is Zilda and I mainly make fashion content, but today I am back with another IB advice video. In this video I just wanted to talk a little bit about how to score a 7 on the IB Biology Internal Assessment. Especially this video I really wanted to make because I ended up spending so much time trying to figure out a bunch of different details about the biology IA that I wish some teacher would have told me. So today we're going to discuss the IB Biology IA. I think some of the information in this video would also be useful if you are interested in writing a biology extended essay. Considering I am a May 2020 student, I did end up getting moderated and marked by an examiner, and I wound up with 23 out of 24 points. So I think I can give some solid advice. I also did some other things which I will post here if you are interested in reading my credentials. You can pause the video now. They're not that interesting. I'm not a teacher, so I also do not take any responsibility if I accidentally misrepresent something. Here's a disclaimer for you to read as well that I will post now. Before we actually start going through my IA, here are the IB marking criteria. If you need further information about the marking criteria, I would recommend you check out the IB website, as they have some good information, or you could alternatively ask your teacher, but considering that you're here, you may not have a great teacher. So without further ado, let's just jump right into structure. Okay, here we have my IA. I'm just going to go through this IA and point out a bunch of different things as we go through. I know this is probably not the best structured video ever, but there's a bunch of minor things I really wanted to point out that could make you lose a lot of marks if you don't do them. So we're doing it this way. I'm sorry. First we have structure. I'm just going to put the structure on the screen here and you can screenshot it if you like. I do want to point out that it is often debated whether the research question should go directly after your title or whether it goes after your background information. And the most logical conclusion I have come to is that it depends on your IA. If you are able to lead your background information up to your research question, I would put it after your background information. However, if it's not, then put it before your background information. Some other general notes are you should not have names, candidate codes, or other identifying factors on your IA. Also, you do not need the date you wrote your IA on your actual IA, and you should definitely always include page numbers to gain full communications marks. Alright, now that we have some basics out of the way, let's start with the title. This should generally be a more concise and general version of your research question that encapsulates the main aim of your research as a whole. Following this, I have my background information. This is pretty much just a summary of the information that is necessary to understand your research question and where it originated. Generally, I would recommend that you start with a short introduction as to why you chose your topic. This should, however, not be longer than about 2 or 3 sentences. And unlike what many teachers recommend, this should definitely not be under a separate section titled Personal Engagement. If you read any of the examiner's reports released annually, you will quickly learn that the IB absolutely hates this subsection and hates long rambles about why you chose your topic, because that is not really what personal engagement criteria is about. So just don't do it. Personal engagement is actually a criteria that is demonstrated throughout your entire lab report, and this is only a minuscule part of how to demonstrate personal engagement. Okay, you can pause the video here, but you can read this statement by the IB basically just talking about personal engagement. I would recommend you read it. It's great. Pause the video. Following the reason you chose your topic, you want an extremely thorough description of pretty much anything that is linked to your research question. Best case scenario, this is structured in a way that leads up to your research question by essentially giving all of the background information on that question first. Since I investigated the effect of lemon juice concentration on E. coli growth inhibition zones, this meant that I wanted to talk about what E. coli are, what exactly is in the lemon juice that causes it to prevent E. coli growth, and the purpose and application of my research. In almost all cases, you will need the purpose and application of your research, because this is just generally linked to what you are actually doing. Like, why are you studying something if it's not relevant, basically? So you want to find somehow that it connects to the real world. So just include that in your background information. The way you organize this background information definitely depends on the flow of your research, but you always want to separate your background information into separate paragraphs to maintain clarity. These shorter paragraphs should revolve around sort of a similar topic that is related to your background information. So the way I started was by introducing the pH scale. Though this is extremely basic chemistry, it is valuable explaining this for biology because I had the space to do so. And it is really not part of the biology syllabus, so it was kind of relevant. However, if I had to remove one section of my IA, this is the paragraph that I probably would have removed. Following this, I explained the common problem that linked to my research question. For my research question, this was related to a disease. So I shortly but thoroughly explained common causes of that disease and the implications. Thereafter, I narrowed down my background information to describe one common cause more thoroughly, namely E. coli, which was part of my research question. This then allowed me to just transition to giving a more biological description of my problem by first talking about the more general aspects of E. coli and then the more detailed ones linking to my actual disease and the research question. From this, we transitioned into the second part of my research question, namely solutions to the problem that have previously been investigated. You should really always include previous research on your topic because that shows that you understand what you are researching, basically. In my case, this was antibiotics being used to treat E. coli infections, and then shortly detailing both biological and also chemical background information about antibiotics because antibiotics are a drug, so chemicals kind of relate to that very closely. Subsequently, I detailed the limitations of antibiotics and proposed my research question and why it was worthy of study, and also gave some background information about lemon juice, which was the thing I was going to investigate. In general, I would say you should probably mention every single part of your research question in some way, especially your compounds that you're mentioning throughout your research question should definitely be somewhere in your background information. In a more general sense, your background information should be structured by stating a problem, detailing why this is a problem, and then proposing a solution and giving general background information on that solution, or depending on your question, simply explaining why your question is still relevant today. To move a little bit away from my own lab and hopefully give you a bit of a more general understanding of what you should do here, we could use the example of vitamin C decay by heat. You want to start off by explaining a general problem linked to vitamin C. For example, you could examine scurvy, which is linked to the biology syllabus. Then you explain why this problem occurs, so we can talk about what vitamin C is and why it decays by heat. And finally, you can explain that this research is still relevant as information about the rate of decay with increasing heat can help potentially find solutions as to how to retain shelf life of products containing vitamin C or whatever else your research links to. The background information section will likely have the most sources in your entire lab because it should be extremely detailed, and it is probably the part of your IA where you want to be extremely careful about plagiarism because a lot of this research, especially in science, is cited off of other people's research. So really do cite everything, even if it's like a small thing, mention it. Be safe. And don't plagiarize. And while we're at it, guys, don't plagiarize my lab, please. That would be great. All right, I trust you guys. Please don't plagiarize. Moving on, we come to the choice of materials section. While officially this isn't a requirement, it aids in maintaining a good structure and is quite a useful section to demonstrate personal engagement. It has also been stated in subject reports that many students benefit from using such sections. Therefore, I would really recommend using this section. What you want to include here is pretty much a description of how you came up with your experimental design and why you chose the materials that you did. For example, I explained I used the top 10 E. coli species because it was the only non-pathogenic species in the laboratory where I did my investigation. This is also the section where you want to describe relevant parts of your pilot study if you conducted one, which you probably should have if you're doing any sort of experiment. These relevant parts tend to be failures you noticed in your pilot study that you adjusted for your final experiment, such as alternative ways of measuring your dependent variable. But you can also point out some things that were good, which is why you continued to use them for your final investigation. This is quite a good place to demonstrate personal engagement because it shows your critical thinking skills and shows that you actually paid attention and realized flaws or strengths of your previous experiment. So really do this section, it's great, guys. As you can see here, I talked about trying to use a glass spreader to distribute the E. coli evenly during a pilot study, but then realized this didn't work and consequently opted to use a different method instead. This is also a good section to demonstrate your safety, ethical, and environmental considerations in practice. Though a full description of these will be made in a separate section later. In this section, you also explain how your IV and DV were chosen and justify why they were chosen. For example, if you were using a range of concentrations, you explain why and how you determined that this range was effective. Typically, this will be determined based on the results of your preliminary experiment. For example, if you took steps of 10, as in 10%, 20%, 30% concentration, and realized that there really was no growth after 50% concentration, you may say something along the lines of we limited our concentration to 50% and narrowed it to 5 steps, as in 5%, 10%, 15% concentration, to notice more changes within a more narrow range, because that would more effectively allow you to research changes in whatever you were researching. I know this section seems kind of weird, but I would really recommend you either read my explanation of the choice of materials or find another IA that has such a section in order to get a feel for what exactly the section aims to do. A lot of the students I've tutored and a lot of the people I know that ended up scoring incredibly well did have a section like this, so I would generally recommend doing this. Next, you explain your hypotheses. This is a relatively self-explanatory self-explanatory section, but a few minor things to point out are both your IV and DV have to be included in your hypothesis. In my case, the IV was lemon juice concentration, or LJC, and the DV was measured growth inhibition zones. Secondly, you have to include the range of your DV if you are using continuous data, such as things like increasing concentrations or temperatures. Include the units and uncertainty of your measured DV. Finally, your H1 should probably also be backed up by a scientific source, meaning a published and credible research paper. I would not recommend using ResearchGate. Use something such as NCBI research articles or Nature papers because those are verified and peer-reviewed journals. So really do your research onto this, because if there's one part of your lab that is quite important for the basis of your entire lab, this is sort of evidence as to why you picked your topic as well. Next, we have variables. You start by naming your IV again, include the range if relevant, and the uncertainty for this. Then you explain your DV and how it is measured, as well as a short description of what equipment you use to measure it. For me, this was a description of what the growth inhibition zone was and how it was measured using a ruler. But if you were measuring something like temperature, it could be as simple as saying the increase in temperature in degrees Celsius using a glass thermometer, or something along those lines. Again, you need the units and uncertainties here though. The final variables are your control variables, and these should always be set up in a table. The table should look something like this, where you have what the control variable is, the reason for controlling the variable, and the method of controlling said variable. As an example, we have the source of E. coli stock solution. As you can see, the reason for controlling here is to minimize the possibility of varying E. coli concentrations of different agar plates, as any errors in forming the stock solution will be present on all plates, thus still allowing comparison of trials. The method of controlling was forming one E. coli stock solution and placing 50 plus or minus 2 microliters from that same stock solution onto the agar plate. As you can see, this entire thing is quite descriptive. You really do want to be quite detailed with everything you write in these things. Note that human rate of reaction will never ever be a control variable, because even if you do use the same person to measure the time taken for every trial, there is an inherent flaw in the rate of reaction of a human, and therefore it will never be perfectly precise, and hence it cannot be used to control the accuracy or reliability of any data. Likewise, your classroom atmosphere or environment cannot simply be said to be a control variable. Firstly, you must specify what specifically about the environment you are referring to if you want to use this as a control variable. For example, are we talking about air humidity, air pressure, temperature, or something else? And also, you have to explain exactly how you controlled it, which is quite difficult for atmospheric pressure or temperature. Staying in the same room the entire time is not a method of control, seeing as airflow can change, altering the temperature of humidity, and various other things can happen. If you are trying to control surrounding temperature or reaction temperature, you could instead try to conduct the experiment in a water bath, for which you maintain a consistent water temperature. Or you could set up a thermometer in your classroom and continuously monitor the measured air temperature. However, I would like to note with this that it tends to be that you will have fluctuations in temperature if you are doing that. In total, I believe you need 8 variables. I'm not sure about that though, but either way, whatever the number was, you want to name 2 variables more than what is required. You do this because even if the IB rejects 2 control variables, you still have the minimum number of required control variables to gain full marks, and consequently, you will not lose any marks, even though 2 were rejected. In contrast, if you are required to give 8 variables and you present 8, there is a high probability that at least one of the variables will not be considered a proper control variable, and so you will easily lose a mark there if you have one eliminated by an examiner. Therefore, just name more than you're required to, unless you really can't think of anything else. Moving on to materials and apparatus. This is quite self-explanatory, but this is just a list of materials and apparatus you used, as well as the quantity or volume you used. Make sure you are consistent in terms of the order, and ideally you also want to name the brand of the equipment or source of materials you used. You also once again need to include the uncertainties of any equipment used. For example, we can see here I wrote 1000 plus or minus 3 microliter Eppendorf pipette with pipette tips. Here the quantity is named first, namely 1 single pipette, the volume and then the uncertainty is named, and finally the brand of the equipment and the equipment is named. As far as I know, there is no required order, except that typically the quantity of whatever material or apparatus is named is usually named first, but even that I don't think is technically required. Also, this is something that should be throughout your entire lab report, but use scientific names, guys. The IB is still frequently commenting that people forget this even when first stating their research questions, so really do include it. And make sure that your species and genus name are in the correct italic or non-italic fonts. Moving on to health and waste disposal. For most people, this would simply be the environmental, ethical, and safety considerations. However, as I experimented on E. coli, this was titled health and waste disposal, which is more important for any E. coli related experiment. The IB has commented several times saying that consideration considerations of safety and ethics were frequently lacking, particularly in work with microbes. To gain full marks for this section, you have to include at least one consideration for safety, ethical, and environmental concerns, and explain how you avoid or minimize the impact of these. You do need one of each though, so at the bare minimum, you should be discussing 3 different things. For example, one that I named was to minimize the possibility of increasing antibiotic resistance, a major ethical concern harmful to the general environment. Very small amounts of ampicillin were used to control, in brackets 10 to 10,000 of ampicillin to deionized water ratio. Here we see the environmental consideration and the way in which we minimize the impacts of using this methodology. This is, by the way, also a great place to demonstrate personal engagement because it shows critical thinking once again. You basically do something like this for each safety, ethical, and environmental consideration. In rare cases, you won't have any ethical considerations. If this is the case for you, which is likely the case if you are running simulations, you simply write that there were no environmental considerations and explain why that is. However, if you're not running a simulation, there's almost always some sort of ethical consideration. Next, we have the procedure, also known as the method. Here, you basically just write in detail what you did. What I mean with in detail here is that you write down literally every single step of what you did. If you dried your test tube with a paper towel, you write that down. If you set your plants in front of a window, you write that down, including the placement as to where you placed it in front of the window. If you tear the electronic balance, you write that down. You kind of get the point here. Also, make sure that you list all equipment you used with specific size if relevant. The only time that it is not relevant what size you used is if there is only one pipette you used in your entire experiment, then it doesn't matter. But as soon as you have 2 pipettes or 2 different beakers, you mention that. A simple way you can check if your method is detailed enough is if you go through your materials list and each single item named there appears in your method somewhere. According to the IB, there is no preference in terms of writing in prose or in recipe style. Nevertheless, I would recommend that you generally write in recipe style because it often forces you to be more concise, as otherwise the list makes little sense. Alternatively, depending on how complicated your lab is, you could write it in a style that is sort of a combination of prose and recipe style, which is what I did. This is a relatively common way to write the method in scientific research. However, this is only really effective if you are able to separate steps into subcategories, such as preparation of E. coli stock solution, preparation of Petri dishes, etc. For most experiments, recipe style should be fine. However, with concentrations, you could totally go and write that in a prose style. Here's also where you again demonstrate safety, ethical, and environmental considerations. For example, in the safety considerations, I mentioned that following the experiment, any areas that were worked on were disinfected using 96% ethanol. In my procedure, I then wrote in step 7, using ethanol and paper towels, all surfaces used were disinfected and cleaned thoroughly. So basically, what you're trying to do here is make your safety, ethical, and environmental considerations reappear in your methodology. If you dilute a solution, it is likely also effective to present that in table format as shown here, since it is simpler to read for the examiner. Following the procedure, you want a diagram of your experiment. Most commonly, this will be a diagram of your setup, or a diagram of how you measured your dependent variable. I don't want to give too much advice here because from what I have read and heard, there seems to be a lot of controversy surrounding what is relevant and what is not. But this image should help further explain your experiment, and therefore it should not be something generic that is simply added for the sake of adding a diagram. An example of being super generic and something that you should not be including is a setup of a burette. This is a very standard scientific setup, so you adding a diagram of that really does not aid my understanding of what you are doing in your IA at all, and therefore adding this is simply wasting space. However, if you measured the length of a leaf, you may want to add an image depicting how you measured the length of a leaf. Did you measure it from the bottom of the stem to the top of the leaf, or did you measure it from the base of the leaf to the top of the leaf? I don't know. That's why a diagram would be helpful. Just like your tables and other figures in your IA, these images or diagrams also always need a descriptive title. It is not enough to say diagram of experimental setup. That is very limiting and doesn't tell me anything about what is in your image. A tip one of my biology teachers said is that if you cover your table or diagram or image and solely read the title of the figure, you should know exactly what is included in it. This works better for tables, but for example, if we cover up figure 6 and read the title, Petri dish illustrating observed E. coli growth inhibition zones for 80% lemon juice concentration, we can predict this image contains a Petri dish with E. coli in it. It also says that the diagram includes 80% lemon juice concentration and a growth inhibition zone. So you can kind of get a visual image of what it is. You should also give credit to any images you took off the internet, and if it is your own image, you can simply state researcher's own image. I'm also not sure if you have to include the date the image or diagram was created, as I've heard conflicting information about this too, but I included it to be safe, though this could potentially inhibit communication marks. After that, you need your raw data table. The title of this is generally written in the format the effect of X
on Y, where X is your IV and Y is your DV. Generally what you want is that your IV is placed on the Y-axis of your table. In my case, that is varying lemon juice concentrations, and the DV is placed on the X-axis of your table. You need to include the units of your IV and DV and uncertainties in the header of each column. Do not put these in each cell, as you are guaranteed to lose marks for communications if you do that. For example, here you can see the title of my dependent variable, the units, and then the uncertainty, which is plus or minus 1 millimeter. I know my uncertainty for my concentrations is not included in this table, however, this is due to the somewhat complicated method I had, so this is kind of unlikely to be the case for you and for most other people, and therefore you are extremely likely to just have your uncertainty there, unlike I did. Convention is also that all numbers should be centralized in tables. That's just standard, and honestly just do it, it's super easy.
Okay, so this video ended up being super long, so I decided to split it into 2 parts, and I will be linking the second video right here, and if it's not linked there, then the video will be coming very soon. I hope this video was helpful, and yeah, have an awesome day guys, and I will see you in the next video if you are interested and have reached this part. Bye!